Quantitative PCR, Session 3, Instrument and Software, presented by Robert O'Brien. In this session, we will be going over the following topics: instrument operation of the AB7500, processing of multi-component data, initial instrument setup, and software setup. The instrument for qPCR is made up of two components: the actual instrument where the PCR reaction is occurring, and a computer which ties into the instrument and instructing it what to do. First, let's go over the basic principle of how the instrument operates. Light hits a tube or a vessel containing the PCR reaction once per cycle. Fluorescent dyes, if activated, emit light corresponding to their spectral characteristics. This emitted light is focused on a detector, and then the computer software interface interprets the detector signal. For the AB7500, the light source is a tungsten halogen lamp. The fluorescent dyes used have wavelengths in the 500 nanometer to 700 nanometer range. There are filters in place, and these allow the light of a specific wavelength onto the detector. And the detector is a CCD camera. CCD stands for Charged Coupled Device. The CCD camera, or Charged Coupled Device, is a light-sensitive integrated circuit that stores and displays data for an image in such a way that each pixel in the image is converted into an electrical signal. The electrical signal is then read by the software. The AB7500 has a thermal block, which is needed for the heating and cooling that is required for the PCR reaction. The filter the light passes through allows for multiplexing of different dyes, as mentioned earlier. For example, the FAM dye for DNA samples and the VIC dye for the IPC. This diagram shows the general schematic of the instrument. The samples are placed on the 96-well thermal block. The light source is directed onto the thermal block through a series of reflections through mirrors. Then the fluorescence given off by the samples passes through an optical filter, and then they are directed onto the CCD camera, where their fluorescence is converted into an electrical signal, which is then read by the software. Now we will go through the PCR detection system in just a little bit more detail. A tungsten halogen lamp directs the light to each well of a 96-well optical plate. Light passes through the optical adhesive cover and excites the fluorescent dyes in the samples. A system of lenses, filters, and a dichroic mirror focuses the fluorescence emission into a charged coupled device camera. Based on the wavelength, the filters separate the light into a predictably spaced pattern across the CCD camera. This diagram shows the approximate layout and position of each of the devices described in the previous slide in the AB7500. For the PCR detection system, during the run, the CCD camera detects wavelengths between 500 nanometers and 660 nanometers from each well. The SDS software, known as the Sequence Detection System software, obtains the fluorescence emission data from the CCD camera and applies data analysis algorithms to it. This chart shows the excitation and emission wavelengths of some fluorescent dyes that are commonly used in qPCR. You will notice that both for the VIC and the FAM dye, which are used in the quantifier system, that their emission wavelengths fall within the range that is detected by the CCD camera. This chart shows the absorption and emission spectra for the 5-FAM dye. The range of light that must be put into the system in order to cause fluorescence is shown in the blue. So the tungsten halogen lamp used in the AB7500 must produce light in this range. The range of light that is emitted from the dye molecule is shown in the pink. So the CCD camera must be able to detect fluorescence in that range. The ability to detect multiple dyes is called multiplexing. If you have a single-plex instrument, for example, you will only be able to detect one dye, for example, FAM. A duplex instrument will be able to detect FAM and VIC, and a triplex will be FAM, VIC, and NED. We will now go on to processing multi-component data. During the multi-component transformation, the SDS software uses algorithms to determine the contribution of each dye. Before the contribution of the individual dyes are assessed, other contributions to the composite spectrum of the dyes must be taken into account. The solid blue line in this graph represents the composite spectrum. The composite spectrum is made up of all the dyes plus the background plus the MSE. An algorithm removes the background component stored in the background calibration file. A background calibration run is done when the instrument is first set up. The background calibration run determines how much background signal the instrument processes during a quantifiler run. Pure dye standards are also run during initial instrument setup. The software is able to use the extracted pure dye standards to express the composite spectrum in terms of the pure dye components. Once the composite spectrum is broken down into its pure dye components, an algorithm applies matrix calculations to determine the contributions of each dye component. This diagram shows how the measured spectrum is broken down into its individual components. The formula for the measured spectrum is given by A plus B plus C plus D plus MSE, where A is the fluorescence of the FAM dye, B the fluorescence of the Vic dye, C the fluorescence of the ROX dye, D the background fluorescence, and the MSE. The MSE stands for the mean standard error, and this indicates how closely the collective multi-component spectrum conforms to the raw spectra. What is the purpose of the ROX dye? The ROX is a passive reference dye. It is not involved in the PCR reaction. It is used to normalize the reporter signal. So how is ROX used to normalize the reporter signal? ROX is a reported dye that is present in every well, so it is exposed to the same conditions as the samples in the well. Therefore, if there are fluctuations in the signals produced by the dyes that may be related to electrical fluctuations, for example, the ROX dye will also experience these fluctuations, which are taken into account when interpreting the strength of the signal, and in turn the quantity of DNA. The SDS software normalizes each reporter signal by dividing it by the fluorescence signal of the passive reference, which is the ROX dye. The Rn, which stands for normalized reporter signal, is equal to the reporter fluorescence divided by the passive reporter fluorescence. Normalizing the reporter signal allows the software to make better well-to-well comparisons of reporter signals. During real-time PCR, the Rn is increasing as the amount of specific amplified product increases. Background is the fluorescence given off during the first few cycles of the PCR reaction. The baseline is set during the first few cycles, usually between 6 and 15. The software then calculates the delta Rn. The delta Rn is the Rn value minus the baseline. The cycle where the delta Rn value crosses the threshold is the CT value. Now let's go into the initial instrument setup. When the AB7500 first arrives in the lab, several calibrations must be done before the instrument can be used to run samples. These are the region of interest calibration known as the ROI, the background calibration, pure dye calibrations, and then an instrument verification run. Even though only a few people will have to be involved in the initial instrument setup, it is important that everyone knows what's occurring during this process. Also, the same calibrations performed during the setup also need to be done as part of the regular maintenance. The first calibration is called the region of interest calibration, the ROI. The ROI is a mapping of the positions of the wells on the sample block so that the software can associate increases in fluorescence with different wells. Basically, you are telling the software the positions of the wells on the sample block. The instrument uses a set of optic filters to distinguish the fluorescence emissions gathered during the runs. The ROI generates a calibration image for each individual filter to account for minor differences in the optical path. It is expected there will be minor fluctuations throughout a plate from well to well. The instrument must know what these fluctuations are, so they are not included in what the instrument is reading as fluorescence from the samples in the well. So basically, the ROI is normalizing the wells. Since you are dealing with very small amounts of DNA, minor fluctuations can change the final quantity readings. The next calibration that is done is the background calibration. The purpose of the background calibration is to measure the amount of background fluorescence in the instrument. The measure of the background fluorescence is important, so in future runs, this value can be subtracted out of the fluorescence signals from the samples. Sources of background fluorescence are background electronic signal, contaminants in sample block, and plastic consumables such as plates and caps. If contamination is detected in the sample block during the background calibration, then the wells where the contamination was identified should be cleaned and the background calibration run again. During runs, it is very important not to introduce any new contamination. This is why it is important that the base of the optical plate is not handled or does not touch the counter surface. Contamination from handling may be incorrectly recorded as low levels of DNA in blank sample wells. The next calibration that is done is the pure dye calibration. The purpose of this is to provide the software with a way to distinguish the fluorescence signals from each dye. This is important because during a run, the fluorescence from all the dyes come off together in a collective fluorescence. The software must have a way to separate out the individual contributions of the dyes in order to interpret their signal and the resulting quantities that will be calculated based on their fluorescence. The pure dye calibration collects spectral data from a series of pure dye standards. It then stores the spectral information in the pure spectral run file, which is a calibration file in the SDS directory. Software can then use the pure spectral data during subsequent runs to characterize pure dyes and distinguish the individual contribution of each dye in the collective fluorescence. A pure dye calibration is done for each dye in a separate plate. The pure dye plates are supplied by Applied Biosystems, and they include all the dyes that are used for sample and IPC detection during a quantifier run. The last part of the initial instrument setup is the instrument verification run. The purpose of this is to test the system after all the calibrations have been performed and before samples and standards are manually set up. This run uses a RNase P plate, which is preloaded with the reagents necessary for the detection and quantitation of genomic copies of the human RNase P gene. The RNase P gene is a single copy gene encoding the RNase moiety of the RNase P enzyme. Each well of the plate used for the instrument verification run contains the TaqMan Universal PCR Master Mix, RNase P primers, a FAM dye labeled probe, known concentrations of human genomic DNA template. All these components are preloaded in the plate, so any problems with the run will be due to the instrument and not due to plate setup, which can happen if the plate was set up in-house. After the run is complete, the SDS software generates a standard curve from the average cycle CT values of the replicate groups of standards. The SDS software then calculates the concentrations of the two unknown sets of samples using the standard curve. Then, it calculates the mean quantity and standard deviation for the different unknown sample populations to assess the instrument's performance. The instrument passes the verification if the analyzed data demonstrates that the instrument distinguishes between the unknown genome equivalents with 99.7% confidence level. Now that the instrument is ready to go, the next step is to set up the software on the instrument. The software must be set up before the instrument can be used to run any samples. The software used in Quantifiler is SDS software. This stands for the Sequence Detection System. This setup should only need to be done one time, and then the information saved so it can be used for future runs. The following must be set up: plate documents and thermal cycle conditions. First of all, what is a plate document? A plate document is a representation of the arrangements of samples, including standards and unknowns, and detectors on the reaction plate. The plate documents are created using the SDS software. The picture at the side shows what a plate document looks like. There are two types of plate documents. An SDS document, which is a .sds file, is the primary file to use when performing a run, and this is required for all experiments. An SDS template, which is a .sdt file, already contains run parameters that are commonly used in plate documents, such as detectors and thermal cycle conditions. A plate document must be set up so that it corresponds exactly to the arrangement of the standards and unknown samples in the reaction plate. Now let's go through step by step how to set up a plate document. To create a new plate document for the AB Prism 7500, go to the AB Prism 7500 SDS software, and once it is open, then go to File New. The next step will be to create detectors, and this is only done the first time the plate document is created. Under the Tools menu of the SDS software, go to Detector Manager, then File, then New. At this point, detectors will be created for human and Y plates. Both human and Y kits use FAM as the reported die. After the information is put in, then click on Create Another, and this will save and set up a new detector. For human and Y detectors, the name must be entered. The die selected is the same, which is FAM. The description can be left blank, and the quench die is left at the default, which is none. A color is selected to represent FAM. The same color can be chosen for both, since one sample will not have both human and Y in the same well. These screenshots from the SDS software show what a completed detector will look like after it is set up, for both the human and the total Y. After human and Y detectors are set up, then a detector is set up for the IPC. The IPC reported die is VIC, not FAM. The quencher die is left at the default none, and the color chosen to represent the IPC is a different color from the color chosen for FAM. At the end of this, click OK to add the IPC detector and to return to Detector Manager dialog box. Here we have a screenshot of a completed detector for the IPC. Notice the name is now IPC and that the reported die is listed as VIC and not FAM, and of course the color here is green, whereas the color that was chosen for the human and the Y was blue. At the end of the setup, the detector should look like the picture below. Notice only one detector, either human or Y, is chosen per well. Once the detectors are created, they can then be added to the plate document. Under Tools, go to Detector Manager, then select Quantify Human, Y, and IPC detectors by clicking them while pressing the Control key. Then click Add to Plate Document. After, click Done. Now that the detectors have been set up, they must now be applied to all the standards. Under View, go to Well Inspector. Then select the wells that the standards will be put into. Most laboratories will always put the standards in the same wells on the plate. Also, all standards are run in duplicate, so the same information must be entered for both wells corresponding to the standards. For setting up the standard wells, select the wells used for the first standard by holding down the control while selecting them. Open the well with the Well Inspector under Views. First, you have to name the standard. These are either done with numbers 1 through 8 or letters A through H are generally used. Then, under the Task menu, use the drop-down menu to select a standard. Then, under Quantity, put in the quantity of the standard. No units are entered, but it is assumed to be nanograms per microliter. Standards are done for human and Y wells independently. So under Task, only select Standard and put in the quantity for human or Y, not both in the same well. The screenshot below shows a completed standard well. Notice that it has a sample name as a Standard A, and that it has a quantity which is listed at 50, which corresponds to 50 nanograms per microliter, which is the concentration of Standard A. Under Task, Standard is listed, and human is checked only, and IPC, not Y. This screenshot shows a completed plate for human only, and it should look like the following. There are a duplicate set of standards, and all the wells are labeled. Now that the standards are set, let's set up the detectors for sample wells. Under the Plate tab, select wells that correspond to where the unknown samples will be. With wells selected, click on View, then Well Inspector. Then, depending on whether the sample is for human or Y, click the appropriate box under Use. Whether it is human or Y, the IPC should always be checked under Use. After this is done, then in the individual wells, put in the names of the samples. The screenshot on this slide shows what a completed well inspector for a sample should look like. Notice that there is a sample name entered, that under the Use column, the IPC is checked and Quantify Human is checked. If there was going to be analysis for Y, then the IPC would be checked and the Y would be checked. Under Task, it is left as unknown because these are not standard, so nothing has to be entered here. And also under Quantity, nothing is filled in because the quantity is what we are trying to determine. This screenshot is of a completed plate document. It shows the first two rows, which are duplicate rows of standards, and then the other rows are for the sample wells. The other wells that are X'd out are because the software does not need to look to these wells for any information, so they are omitted from the plate. After the plate document is set up, the next thing that needs to be checked is the thermal cycle conditions. Before using the AB7500, two changes must be made to the thermal cycle conditions. The thermal profile and the sample volume must be adjusted. First, the thermal profile. The first stage in the thermal profile is the hold stage, and this must be deleted. The sample volume should be changed to 25 microliters, and the 9600 emulation should be checked. By checking the 9600 emulation box, you're reducing the amp rate. To adjust the thermal cycle conditions, under the Instrument tab, press the Shift key and click on Stage 1, which is the hold stage. After the hold step is selected, press the Delete key. After this is done, the first stage should be 1 rep at 95 degrees Celsius for 10 minutes. This is the same step in hot start PCR, which is used to activate the Amplitech Gold DNA polymerase. This is a screenshot showing the correct thermal cycle conditions. Notice that stage 1 is set at 1 rep at 95 degrees Celsius for 10 minutes, and that even though it is grayed out, the sample volume is set at 25 microliters, and that the 9600 emulation box is checked. After the plate document is completed, it is saved as a .sds file. Under File, go to Save, then select the location where it will be saved. Enter a file name and then Save as Type you select SDS document, which is a .sds file. Then click Save. Template files are generic plate documents that are set up to ensure uniformity in the laboratory. Most laboratories will set up template documents so that everyone is using the same plate document when running their quantifiler. The only information that would be changed from one plate document to the next would be the names of the samples and whether they are for total human or Y quantitation. Template documents contain the following information: assay-specific detectors, well assignments for quantification standards with detectors, tasks, and quantity already filled in, well assignments for unknown samples with detectors already set, so only appropriate assay needs to be checked, instrument settings to include thermal cycler and reaction volume settings. Template documents are saved as a .sdt file and saved under Templates. This is a screenshot of a completed template plate document with the wells for the standards already filled in. There are two sets of standards. One set would be for human and the second set would be for the male standards. Here we have a screenshot of a completed well inspector in a template file. Notice that the sample name is already filled in, the quantity is already filled in, under task, standard is there, and it is checked for quantifiler human and IPC. That means this is a well inspector for a human standard. This is the end of session 3.